going to be taking you through cell-mediated immunity. I understand that last week, this week, it hasn't ended. On Wednesday, you had a class with Fiona on Wednesday where you studied humoral immunity. So you basically studied about the antibody-mediated immunity. And on Thursday, you studied vaccines with Jackson Were. And on Friday, you managed to study complement with Dr. Kabanda. So basically, today I'm taking you through cell-mediated immunity. Why am I giving you a recap? The same way we saw innate immunity the other week, that innate immunity has a couple of mechanisms that govern it for it to elicit an immune response. That is the same way with adaptive immunity that we started on Wednesday. It has a couple of mechanisms that govern it. And among these mechanisms, we get to meet the humoral immunity, which is basically the antibody-mediated immunity, which I'm so sure Madam Fiona took you through so well. And then the other aspect, the other mechanism is the complement system, which I'm so sure Dr. Kawanda took you through so well. So the last component of it you're going to be studying, the ones that govern it, is the cell-mediated immunity. The other things that are going to be coming in, that you're going to be studying in the coming lecture, basically the challenges the immune system faces, and you end up with complications. Okay, so when I talk about cell-mediated immunity, what comes into your head? I think I'm going to need a pointer for this. Okay, so basically cell-mediated immunity, we're talking about the T-cell immunology. That is the T-cell immunity. That is the CD4 and the CD8 T-cell immunity. So within the adaptive immune response, we get to see that there's actually cooperative interaction of both antibody and cell-mediated immunity. And this cooperation provides the best opportunity for combating infection. We're actually going to see, you've already noticed that an effective antibody immune response is achieved by activation of T cells. I'm trying to explain to you how antibody and cell-mediated immunity work together to achieve an immune response. We are going to see that they work hand in hand. Basically, we get to see that we're even going to see ahead that when T cells are activated, they go ahead and activate the maturation of B cells. See a B cell turning into an effector cell. We're even going to see that specific T cells, when they're also activated, they also activate the B cell to be able to switch to another immunoglobulin class. So basically, antibody-mediated immunity and cell-mediated immunity are units that work together to achieve the immune response of adaptive immunity. So for the sake of today's lecture, we are going to be focusing on T-cell mediated immunity or cell-mediated immunity, where we're going to drive our attention to where the T cells are activated and get to see also how these cells are able to recognize antigens. So we're going to dig further and get to see. We're also going to look at the different T cell subsets and their functions as well. And we're also going to look at the T cell development, proliferation, and differentiation, which is going to be a little bit of a repetition because we've seen this before. So let's start off T cells. Just a minute. I hope I'm not interrupting you. Okay, so let's start with the development of T cells. Okay, I need to hear from you guys. Just a minute, just a minute, just a minute. Are we all together? Can you see my screen?
Yes.
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 Yeah.
Okay. And am I fast before I proceed?
Yes.
Fast.
You're
But
not.
you're breaking.
Okay. Okay. Cool. Let me try and see how I can. I hope I'm loud enough for everyone. I'm going to leave your microphones on, but please turn your microphones off. I'm going to leave the option of you being able to unmute in case you can't hear me, but keep your microphones off so that we can run smoothly. It's a short lecture. So I said we're going to start with the development of T cells. So how do the T cells actually develop? I'm going to give you a small story back in the first lecture that we had. We got to see that all these cells are formed from the hematopoietic stem cell. It's basically a recap. Where we get to form different lineages, the myeloid lineage and the lymphoid lineage. The myeloid lineage or myeloid progenitor is where we get to see the cells that take role in innate immunity, where we get to see the phagocytic cells. We go to see all that. It's under the myeloid lineage. The lymphoid progenitor or lymphoid lineage, this is the one that differentiates to form the large lymphocytes, which differentiate further to form the natural killer cells, which we saw that they take role in innate immunity. Differentiates to form the large lymphocytes, those natural killer cells, and the small lymphocytes, which are basically, they differentiate to form the CD4 and CD8. Those are the ones, no, and the small lymphocytes. So it's the lymphoid progenitor dividing from the large lymphocytes that differentiate to form the natural killer cells, and small lymphocytes differentiate to form the B cells and the T cells. But for the focus of today's lecture, we're going to be looking at the T cells that differentiate further to form CD4 and CD8. But let's see, how does this happen? So basically, T cells are derived from the same hematopoietic stem cell as the B cells. However, we get to see that the B cell maturation continues in the bone marrow. On the other hand, the T cell maturation continues in the thymus, where they mature and undergo differentiation under the influence of thymic hormones. With time, we get to see that the T cells differentiate into committed cells expressing both a specific T cell receptor. We're going to see what a T cell receptor is on a T cell and what its role is. We're going to dig this into, we're going to dig further into this. So as these T cells are undergoing prolific processes, that is the positive selection and negative selection. So what happens in the positive selection is that these T cells are separated on whether they're able to identify self and non-self. So the cells that recognize self class 1, let me give you a recap on this before I proceed. Remember, we said that all cells in the body express a major histocompatibility of class 1, except the antigen-presenting cells, which are the dendritic cells, the B cells, and the macrophages. These ones, they express MHC1 and MHC2. Now, for the cells that express MHC1, the body, they use it for the other phagocytic cells and other cells of the immune system to be able to differentiate between a normal cell and an infected cell. On the other hand, the antigen-presenting cells that have MHC1 and MHC2, they use it for presentation of the antigen to CD4 or CD8. So during the positive, the cells that recognize self-peptide are self-MHC. That means the cells that recognize MHC class 1 on a normal cell with weak affinity, they survive. They are not killed. And they are termed as self-MHC restricted. The body can proceed with those cells to maturation. Since the body is now sure that those cells, those T cells, will not attack cells. That is positive selection. The body is literally selecting those that will proceed in the immune defense. In negative selection, the body is literally eliminating those cells that recognize the self-peptide plus the self-MHC1 with high affinity. Meaning as these cells are maturing, they still have that confusion. So the body wants to know, as you're maturing, are you able to recognize the self-MHC1. And if you're not able to recognize it and you attack it in negative selection, these cells are killed. So that is still under the development of the T cells. Those are the big processes they go through. So once they've undergone these processes, the survivor cells, that is the cells that have been picked in the positive selection, are this CD4, CD8 double positive T cells, and they continue to mature into A cells. What do we mean by this? Let's run back to the lymphoid, and we are focusing basically on the small lymphocytes, the ones that differentiate from the B and the T. Let's lymphoid progenitor, the one that differentiates to form the cells. Initially, these cells differentiate into different cells carrying initially, because these cells are still immature. These cells are divided to form T cells that carry both CD4 and CD8 glycoproteins. That's them double positive T cells. That is initially, because when they're still naive, when they're still immature, the lymphoid progenitor divides to form these cells, CD4 and CD8. So in the body, you're having them circulate the CD4, CD8. However, when these cells encounter an antigen presenting cell, remember an antigen presenting cell, like I said earlier, has MHC1 and MHC2 peptides on its surface. I'll give you a reminder on this. Remember, CD4 cells bind to MHC2 and CD8 cells bind to MHC1. So we're literally having an antigen presenting cell, an example being a macrophage. So we're having an naive cells carrying both CD8 and CD4 glycoproteins on the cell. And we're having a macrophage or antigen presenting cell carrying both peptides, MHC1 and MHC2, right? So when this double positive T cells, any in circulation, they're still naive. When they encounter a macrophage carrying both peptides, MHC1, MHC2, you're going to find that each of these double positive T cells will attach to one side of the macrophage with a specific receptor. So you can find that one of the CD4, CD8 double positive T cells may attach to one side of the macrophage, maybe MHC2, but if it attaches on MHC2, it will use the CD4, meaning it is leaving one side open of the macrophage, and that is the MHC1, meaning another naive cell that is in circulation, a naive double positive cell in circulation, CD8 glycoprotein. Now, remember, these are the survivor cells that pass the positive selection. Now, once they've attached to this antigen cell, sheds into mature cells, either CD4 or CD8, meaning the CD4 that is MHC class 2, and it recognizes that antigen, it will lose its CD8 and will remain as a CD4 cell. Then the CD4, CD8 cell that attaches on the MHC1 using the CD8, it will lose the CD4 and remain as CD4 T cell. I hope that is clear. So we get to see that all these processes, only a minority of developing T cells express appropriate receptors to be reached the mature T cell pool. So basically, this is development. I was telling you what actually happens. Now let's proceed and get to see the T cell receptor for antigen. It's known as the TCR. So what is a TCR? Basically, each cell, whether CD4 or CD8, microphones off once again, I beg. Microphones off. Okay. So each cell, whether CD4, each T cell, whether CD4 or CD8, they all have a T cell receptor. And what is the role of this T cell receptor on a T cell? We get to see that the T cell receptor helps in the recognition. It is the recognition molecule of T cells. It helps them recognize antigens that are bound on the antigen-presenting cells. But what is interesting, we get to see that this T cell receptor is divided into different classes depending on the subunits it could possess. This T cell receptor could possess the alpha beta subunit or gamma delta subunit. I remember about gamma delta subunit, the T cell receptor in innate immunity. Let me first give you an example of the alpha beta subunits for T cell receptor. We basically have a T cell receptor that is helping a T cell recognize an antigen. But then the subunits of one that differentiates this alpha beta TCR receptors. They can have either CD4 or CD8 markers. However, little is known about the gamma delta T cells. But the little information that is known about them is that they fall under innate immunity, and something in they don't need an antigen presenting cell, neither do they need MHC1 or MHC2. However, when they are infected, we get to see that the protein fragments of the pan.
Madam Barbara.
Yes, please.
Your network is completely breaking. Yes, but your network is completely breaking.
I'm so sorry about that. So where did you start hearing me from?
No, we are hearing, but there's a lot of interruption.
Okay. Okay, but are you with me? Are you still following? Are you still following despite the
Yes, yes, ma'am.
Okay, let me just try and it could be, I don't know, but looks like my Wi-Fi is stable. Let me just try my best. If you can't hear me, please warn me as quickly as possible so that I don't lose you. So I was telling you, these T cell receptors, right? They can have different glycogens or surface markers, either CD4, these can have different subunits, either alpha, beta, or gamma delta. And we had said that the gamma delta subunits of a T cell receptor, normally the examples of those are the CD4 and CD8. But the gamma delta subunits of a T cell receptor, those are the cells that you find in innate immunity. They don't play a role in adaptive immunity. And for them, they don't need an MHC1 or MHC1 antigen presenting cell. They will just effect an immune response by binding the antigen one infected cell. So I was telling you that T cell receptor, whether alpha, beta, or gamma delta, both have a variable region, constant region. Now, the constant region is the one close membrane, like I said earlier, while the variable region is the one that binds to the peptide A complex. So I was trying to bring more light on this. I was giving, however, when they are infected, we get to see that the protein fragments of the pathogen tend to form a complex with the MHC class 1. That complex becomes an antigen peptide MHC1 complex. It's basically the protein fragments MHC1 to form antigen peptide MHC1 complex. Now that complex has a different structure from the MHC1. So when a phagocell or an immune cell comes towards that cell, they're able to differentiate it from a normal cell. They can actually tell it's infected. So when a T cell approaches the infected cell, it uses the variable to attach to either the peptide MHC complex, that is the MHC1 if it is normal, or the antigen peptide MHC complex when the cell is infected. And when this T cell receptor interacts with the, you see that a series of biochemical events occur that will lead to initiation of functional activations of T cells. Now, presenting cell that is having a complex on its surface, the antigen peptide MHC1 complex. And we're having either CD4 or CD8. All of them have T cell receptors. So I'm going to start with that T cell receptor. T cell receptor that interacts with this antigen stimulator. We get to see that on top of the TCR complex is required, and this signal is given by the CD4 and CD8. Remember, we've been talking about the T cell receptor and talk about CD4 or CD8. So if a CD4 T cell, you have a question. Can you guys hear me? You guys hear me?
No, it's very bad.
Okay,
you are on and off.
I think it's basically the weather. Just 1 minute. I'm connecting my modem, so whoever is not hearing me for now, it's not your network, it's me trying to connect my modem so that we are on the same page. I hope I'm loud enough now. You guys can hear me now? Can you hear me? Can you hear me? Can you guys hear me?
Yes, we can.
That is good enough. So can you see my screen share? Can you guys see my screen share? Someone should respond. I need to know whether we are together or not. Can you see my screen share? Okay.
Yes, we can.
Okay, so I was telling you, I missed that from somewhere a bit. So I was telling you, basically, we basically have an antigen presenting cell. We had talked about the first signal. Let me repeat about the first signal. We have an antigen presenting cell expressing the antigen peptide MHC complex on its surface. And it encounters a T cell, either CD4 or CD8. What is interesting about these T cells, whether CD4 or CD8, they all carry a T cell receptor, and it's the one that makes the first signal. So the T cell receptor of either CD4 or CD8 will interact with this antigen peptide MHC complex. After it has interacted, this is not enough to stimulate the T cell. We're going to see that a second signal is required, which is given by either CD4 or CD8, depending on the cell that was encountered. If it was a CD40 cell, we're going to see that the T cell receptor would interact with the antigen peptide MHC2 complex. And after that binding of the complex with the T cell receptor, a second signal is required from the CD4 glycoprotein, which literally comes and attaches to the antigen peptide MHC class 2 complex. It just attaches to it. The role of this CD4, it's basically confirming a firm grip of the antigen presenting cell onto the T cell. That is why we call it a second signal. It's ensuring that the antigen presenting cell and the T cell are firmly held together. On the other hand, if it was a CD8, meaning we basically have an antigen presenting cell presenting the antigen and the antigen peptide MHC class 1. This interacts with the T cell receptor of the CD8. And then the second signal would be given by the CD8 glycoprotein attaching to this complex, ensuring that the antigen presenting cell and the CD8 T cell are held firmly, right? So this is what I was telling you here. The CD4 and the CD8 molecules on the T cell function as co-receptor molecules. Now, CD4 binds to MHC2 molecules and CD8 binds to MHC1 molecule. Let's proceed and get to see where the story goes. So basically, we've seen that one happens, but that was literally a summary. Now we are going to see in particular what happens to CD4 and what happens to CD8 when they encounter an antigen presenting cell, presenting this antigen on the surface. So basically the T cell proliferation or multiplication depends on a series of events. Let's begin with MHC class 2. So we're going to be focusing on the CD40 cells. Let me give you a summary of this without reading so that you can move with me. So basically, this is what I was talking about. The first signal. I need you to note from the start that the CD40 cells kind of differ from the CD8 cells. We're going to see how they differ. Their proliferation differs. You're going to appreciate by the time we finish. So basically, the CD40 cells need a first signal and a second signal. So what is in the first signal? When an antigen presenting cell approaches a CD40 cell, it's expressing the antigen on the MHC class 2. And it's presenting it as the antigen peptide MHC class 2. I hope we are together. So the T cell receptor of the CD40 cell is the one that interacts with this complex, the antigen peptide MHC2 complex. Now, still in the first signal with the CD40 cell, still in the first signal, we get to see the CD4 glycoprotein co-receptor binding to this complex on the MH2. So it's literally the T cell receptor binding to the antigen peptide MHC2, and CD4 glycoproteins come and bind to this complex to ensure that the antigen presenting cell is having a firm grip with the T cell. However, this is what I was saying, that this binding of the CD4 glycoprotein helps to ensure stability between the T cell and the antigen presenting cell. However, this is not enough for the CD40 cell activation, which is different from the CD8 cells that we have seen. This is not enough. This is the first signal, but it's not enough. So what happens? They require a second signal to actually activate these CD40 cells. Where does the second signal comes from? This one is derived from the interaction of the B7 family for stimulatory molecules, that is CD80 and CD86. These are present on the antigen presenting cell. They appear on the antigen presenting cells as ligands. So basically the antigen presenting cells has other ligands. It has CD80 and CD86. And these ligands interact with a ligand on the T cell known as CD28. Now, both of them don't have to interact with this ligand. Either of them can interact with this ligand. So as long as we've had the first signal, that's the T cell receptor combining with the complex, and the CD4 glycoprotein combining to this complex to ensure stability. Then a second signal will come in where the antigen presenting cell has ligands, CD80 and CD86. So either of these ligands then binds to CD28 on the C cell. It could be CD80 and CD28 or CD86 and CD28, but this doesn't happen unless this happens. So literally one process stimulates another to occur. So when CD4 has ensured stability between the T cell and antigen presenting cell, we get to see that a second signal is stimulated to occur. That is when either of these ligands, either CD80 will combine to CD28 on the T cell, or CD86 will combine to CD28. Now that is a second signal. Now that is effective. Let's see what happens when that signal has been given. Now, upon completion of these two stimulatory steps, that's the primary and secondary, we get to see a set of biochemical pathways triggered in the cell that result into multiplication of the T cell. However, in these biochemical processes, when these primary and the secondary activations have occurred, we get to see a couple of events occur, and among these events, we are having the T cell that is being activated, releasing cytokine, right? We want to see, I'm taking it back, we want to see how the T cell gets to stimulate an immune response. So once the activation has been successful, we get to see a couple of reactions occur that enable this T cell to secrete cytokines. Remember, cytokines are signaling molecules in the body. Among these cytokines, we get to see that T cell secreting interleukin-2. This is an interesting statement I want you to be with me. We get to see the T cell, the CD4 T cell secreting interleukin-2 and interferon gamma. Interferon gamma is antiviral and antibacterial. Interleukin-2, I'm going to explain on it, I'm holding on it. So you get to see that T cell is secreting interleukin-2, interferon gamma. And we get to see even that T cell increases the expression of interleukin receptors. What I was saying. So basically, the reason I said is a couple of biochemical pathways are triggered. So remember, we had a T cell that encountered antigen presenting cell, and we had the primary and the secondary signal, right? After that signal, that signal is telling the antigen presenting cell and the T cell, yeah, this has been complete. Now an immune response has to be stimulated. The antigen presenting cell secretes a cytokine. Remember, cytokines are signaling molecules. It secretes a cytokine, in particular interleukin-12. Remember, this is produced by the antigen presenting cell that presented the antigen in the first place. So it secretes interleukin-12, and interleukin-12 stimulates the T cell to secrete interleukin-2. Now, before I proceed, that process whereby the antigen presenting cell is producing interleukin-12, that is stimulating the T cell to produce interleukin-2. That process is known as paracrine signaling. You're having a cell that is producing a signal that is effecting on the neighboring cell. Now let's see what happens to the T cell when it's signaled to produce interleukin-2. Now this interleukin-12 that was released stimulates the T cell to produce interleukin-2. At the same time, also stimulates the T cell to produce receptors on its surface for interleukin-2, which is funny. I'm being signaled to produce interleukin-2 at the same time. I'm being signaled to make receptors on my surface for interleukin-2. So what happens? The interleukin-2 that is being secreted by the T cell affects on that same T cell on the interleukin-2 receptors. So interleukin-12 came and signaled me to produce interleukin-2, and also signaled me to put express interleukin-2 receptors on my surface. And when I started producing my interleukin-2, it came and attached on my surface on the receptors I made and affected on me. So basically, the T cell is producing signals that are working on itself. It's producing cytokines that are working on itself, and that process is known as autocrine. I hope we are together. The cell is producing signals that are working on itself. Paracrine, we got to see the antigen presenting cell secreting interleukin 12 that affects the T cell to produce interleukin 2. That is paracrine. On the other hand, autocrine, the T cell is producing interleukin 2 that affects on the same T cell. What does interleukin 2? What does it do? It favors this maturation of the T cell. And remember, this T cell is producing interleukin 2 that is favoring its maturation. Also, it's producing interferon gamma that is antibacterial and antiviral. So basically, a couple of things are happening that favor this T cell to kill the pathogen. We're also going to talk about, this is talking about the T cell receptor. Now we know that the T cell is activated. The next slides are going to tell us when the T cell is activated, what does it do? This is giving you an overview of what is coming ahead, right? So these T cells are able to proliferate and differentiate into effector cells. So this has been a process. It gets the first signal, the second signal, and when it receives the second signal, so we basically had the primary signal. You could remember that has the first signal, second signal, that's a CD4. Let me give you a summary before we go to CD8. So we have an antigen presenting cell that basically presents the antigen and the antigen peptide MHC class 2. This interacts with the T cell receptor on CD4, making the first signal. Still in the first signal, we get to see the CD4 glycoproteins binding to this complex to ensure a firm group of the antigen presenting cell and the T cell, right? However, that is not enough for the T cell. We get to see it requires co-stimulatory molecules. Those are the ligands that are present on the antigen presenting cell and the T cell. Am I losing someone? Okay, I was giving you a recap. So basically, in the second signal, we get to see the CD80 ligand and the CD86 ligand present on the antigen presenting cell. Either of these can bind to the CD28 ligand present on the T cell, and the second signal is fired. Once the second signal is fired, we get to see a couple of biochemical pathways triggered. In these pathways, we get to see the T cell being triggered to produce interleukin 2 by the antigen presenting cell. Remember, the antigen present after the first and second signal, it secretes interleukin 12 that stimulates the T cell to produce interleukin 2, which affects on the same T cell using interleukin 2 receptors, a process known as autocrine. And like I said, interleukin 12 affecting on the T cell, causing it to produce interleukin 2, that is paracrine. On the other hand, we get to see even the T cell producing interferon gamma. Now we are going to see ahead and get to see, yes, they've produced this, but what else happens? We are going to dig further into that. Now let's also look at the CD8 T cell activation. And we also come to the same level we've reached with the CD4 T cell. Then we can proceed and get to see what these effector cells, now that T cell has been activated, but then once it's activated, what does it do? That is going to be our next slide. Now let's see how the CD8 T cell is activated and get to see how does the CD8 T cell activation differ from the CD4 T cell. So now I'm going to give you another story again. So basically, when an antigen presenting cell, presenting the antigen on MHC class 1, remember MHC class 1 presents to CD8. So basically, you're having an antigen presenting cell, which is sick with or infected with the pathogen, meaning on its surface, it's presenting a complex known as the antigen peptide MHC1 complex. Let's move together. So this antigen peptide MHC1 complex interacts with a CD8 T cell on its T cell receptor, right? Which is the first signal. This was also a similar signal in the CD4 T cells, but we want to see where the difference comes in. Now remember, this signaling is not enough. We get to see the CD8 glycoprotein the same way we saw the CD4. Now the CD8 glycoprotein comes and binds to this complex of antigen peptide MHC class 1 with the T cell receptor. It binds to it. It has the same role like the CD4. It's ensuring that this complex on the antigen presenting cell is bound tightly on the T cell, right? So again, this interaction keeps the two cells bound together during antigen specific activation. Now, there's a story you're not going to hear. You're not going to hear about the other co-stimulatory molecules when we talk about the CD. Binding to this complex, now the CD8 T cell is already activated. It will start producing interleukin-2 the same way we saw with the T cells. So basically for it, when the complex, the antigen peptide MHC class 1 with TCR complex is formed, that's the first signal. Their second signal of CD8 is when the CD8 glycoproteins bind to this complex to ensure a firm grip. After that, the antigen presenting cell will stimulate this CD8 cell to produce interleukin-2 the same way it did in the CD4 cells. And this interleukin-2 will effect on the CD8 cell and ensure its maturation. At the same time, we get to see the CD8 T cell producing interferon gamma that has antiviral and antibacterial processes. And at the same time, we get to see the CD8 T cell producing growth and differentiation factors for T cells. So basically, when it's activated, whatever cytokines it produces stimulate its growth into effector cells. Now, I want to note out the difference between the CD4 and the CD8. Unlike the CD4 cell activation, CD8 T cell activation in most cases is independent of co-stimulation. Remember, with CD4, we needed co-stimulation from the ligands, CD80 and CD86 interacting, either of these interacting with CD80, CD86 on the antigen presenting cell, giving a second signal where they interact with the ligand CD28 on the CD40 cell. That is a second signal of the CD40 cell activation. On the other hand, the CD8 don't need those co-stimulation molecules. For it, as long as the T cell receptor binds to the antigen peptide MHC class 1 complex and CD8 binds to this, that is enough. The CD8 T cell is activated. More so, the virus-affected cell is destroyed through cytotoxic granules released by CD8 cell. This we shall dig further into it in the next slide. For the CD40 cell, we get to see that when it's activated, it stimulates other immune cells, come to the site of infection to kill the pathogen. On the other hand, when the CD8 cell is activated, for it, it's ready to destroy the pathogen. The effector cell now migrates into the body. When it encounters the pathogen, we're going to see it degranulating. Within the CD8 T cell, the slides ahead are explaining it, but I'm giving you an overview. The effector CD8 T cell, that is the one that is mature. It contains cytotoxic granules. These granules have activated proteins that are toxic. I'm going to talk about them later. That's perforin, granzymes, and granulocytes. So what happens when this CD8 T cell encounters a pathogen? It degranulates. It literally opens up and releases these granules to attack the pathogen. That these granules that kill the pathogen, like we shall see in the slides ahead. Now, this is different for the CD40 cells. They don't do that. Instead, they bring other cells to the site of infection. So let's see, what are the functions of the CD4 effector T cells and the CD8 effector T cell? Let's start with the CD4. So basically, the maturing CD40 cells can become, when they've been triggered after the first and second signal, they can differentiate to form the TH1. They can differentiate from basically one of the four main categories depending on the infection. The assignment I want to give you, I necessarily didn't put for you the infections each of these cells kind of, normally you can find that these T cells are triggered, a T cell is triggered to maturate into either TH1, TH2, TH7 regulator cells. Depending on the infection, I want you to go ahead and know, research for me and get to know when are the TH1 cells formed, in which kind of infections? When are the TH2 cells formed, in which kind of infections? I want you to find out that for me. It's a bit very easy, but it will help you in the reading. So let's see the Th1 cells. What are they? How are they formed? So basically, this Th is T helper cell. So when a T cell is activated, it can actually differentiate into either of these cells depending on the infection. But also, normally the infections present in the body trigger presence of certain cytokines, signaling molecules that help the T cell to differentiate into that particular T cell. For example, we get to see that, sorry about that. We get to see that Th1 cells are triggered to form in presence of when the environment around them has interleukin-2 and interleukin-12. I hope we are together. If the surrounding of the T cell, there is interleukin-2 and interleukin-12, a T cell can actually effect to form a T helper cell 1. Now, when this T helper cell 1 is formed, it has different roles from all the other different categories of the T cell. For it, it activates macrophages or B cells. It activates macrophages. And when macrophages are activated, the same way we discussed in innate immunity, when they're activated, these are phagocytic cells. They will engulf the pathogen, and we got to see the mechanisms how these phagocytes actually get to kill the pathogen. We got to see that within the phagosome there is acidity. All that we covered it in innate immunity. So macrophages are activated that come and kill the pathogen directly, or by producing interferon gamma, which can also kill the pathogen. Interferon gamma, once again, it's antiviral and antibacterial. Mostly we get to see when the T helper cell 1 is activated, on top of activating macrophages, it also activates B cells to switch to produce different subclasses of immunoglobulin G. I'm so sure that lady that through humoral immunity should have covered this. I hope she did. So now that we're seeing a T cell, that's why I was telling you that humoral and cell mediated actually work together. We're seeing a T cell activating B cells to change an immunoglobulin class to another class, depending on the infection. In either case, we get to see this can promote bacterial clearance, either by direct destruction in interferon gamma activated macrophage or by destruction after phagocytosis of opsonized particles. I had told you this. So whatever the T helper cell is secreting aims at eliminating the pathogen. Then T helper 2 cells, for them, they are triggered to form in an environment where we have interleukin-4 being produced. In presence of this interleukin, we form Th2 cells. What do Th2 cells do? They predominate and activate mast cells and eosinophils. Something about are you hearing me? Are you hearing me? Can you hear me? Can you hear me? Just a minute.
Yes, we can hear you.
Okay, I just want to be sure that I haven't lost you. That would be bad. So something about the Th2 cells, we've seen that when they're activated, they activate mast cells and eosinophils. And they also activate B cells to synthesize immunoglobulin. Let me give an explanation here. You remember mast cells and eosinophils when we were talking about innate immunity. Mast cells and eosinophils are chemical secreting cells. They secrete histamine. And what does histamine do? It's a mediator of inflammation. It causes the epithelial cells that line the capillaries to widen a bit, which results into enlarged capillaries or wide capillaries. When these capillaries are wide capillaries, which initially shouldn't have been penetrated, we get to see phagocytic cells coming inside these capillaries. We get to see red blood cells. We get to see interstitial fluid. Everything piles in there, and all this results into an inflammation. That is what mast cells and eosinophils do. On the other hand, these T helper cells to also cause B cells to synthesize immunoglobulin E. I'm so sure the lady that took you through B-cell immunology should have taken you through immunoglobulins. Immunoglobulin stimulates mast cells and eosinophils to secrete. It actually stimulates mast cells to secrete histamine and also stimulates the production of eosinophils. So basically, everything that these T helper cells does, it stimulates inflammatory responses. It mediates inflammation, and all these processes lead to a response to helminth infection. This was I cheated this for you. I wasn't meant to tell you Th2 cells are associated with helminth infections. I wanted that to be your assignment, but we get to see it helps in the response to helminth infection. More so, these T helper cells, when they are activated, when they're formed, they also produce other inflammatory cytokines like interleukin 4, 5, 9, and 13. So you're seeing how the T cells affect their functions in different ways. Let's look at Th17. Now this one is formed in presence of transforming growth factor beta, interleukin 6 and 23. When they are present, we get to see T cells differentiating to form Th17. When Th17 is formed, these cells produce cytokines. Cytokines, you'll find them all over because they're signaling molecules. Among the cytokines Th17 produces is interleukin 17, 21, and 22 that have different roles. I gave you an example of 17. Let's see how 17 helps in mediating an immune response. 17 induces the stromal and epithelial cells to produce interleukin 8, which is a potential cytokine in recruitment of neutrophils and macrophages to infected tissues. And as long as you see the neutrophils and macrophages come into an infected tissue, then they will phagocytose them. More so, macrophage will go ahead to present this antigen to other cells. So basically, it's one unit. Then the last of them is the T regulator cell. Now, these ones are formed in presence of only transforming growth factor beta. As long as it's present in circulation, a T cell will differentiate to form a T regulator cell. What do they mean? What do they do? The same way you hear the name T regulator cells, they regulate the function by suppressing T cell responses. They literally tell the T cell, calm down. That is enough. You can now stop. They regulate the T cell responses. And how do they do that? They produce, after being stimulated to be formed, they themselves start producing transforming growth factor beta, interleukin 10, which can suppress the immune responses of the T cell. Now, let's look at those were the CD4 T cells, CD4 effector cells. And we've seen how they can differentiate into different types of T helper cells and what the different types of T helper cells can do. And I gave you an assignment to help me find out those different types of T helper cells that are formed in which kinds of infection. I cheated for you by telling you one of them that we get to see in, okay, let's proceed. So let's look at CD8 effector cells. Let's get to see what do they do when they've turned into effector cells. So now, CD8 cells differentiate into effector cytotoxic T cells by engagement of the T cell receptor and recognition class of MHC1 peptide complex on the surface of an infected cell, right? We talked about that, the same way we did with the CD4 T cell. The antigen-presenting cell with the antigen peptide MHC1 complex interacts with the T cell receptor of the CD8. We get to see another signal given in, a secondary signal given in by the CD8 attaching to this complex to ensure a firm grip. And when this is complete, we get to see that CD8 is actually stimulated to become an effector cell. But among those processes that stimulate after that first and second signal, the antigen presenting cell produces interleukin 12 that makes the CD8 T cell to produce interleukin 2, which acts on itself, stimulating its maturation into CD8 effector T cell. Now, when that effector T cell has been formed, it moves in circulation. And when it encounters an antigen, following recognition of this antigen, the CD8 T cell proceeds to kill the infected. And how does it do that? How does it do it? Like I told you earlier, the method of killing is through cytotoxic granules. Unlike the T cells that signal other cells to come to the site of infection, they signal other signaling molecules to signal other cells come to the site of infection to clear the pathogen. The CD8 T cells are self-contained. They encounter the pathogen and kill it there and then. How do they do it? They use cytotoxic granules. They basically have granules that have activated proteins, that is perforin, a family of granzymes and granules. The perforin helps the granzyme and granulosin to enter the infected cell. And once inside the infected cell, we get to see the granzyme and granulosin initiating apoptosis, programmed cell death, by activating cellular capsaicin. So similar process occurs when with CD8 cell recognition of tumor cells. So this is how a CD8 interacts with a pathogen when it encounters it. I think we've really summarized. That was basically about the T cell mediated response. Now I want to hear questions from you. Did I confuse you? Did you understand what exactly happened? Can I hear questions from you, please, if you have any? Do you have any questions? Can you guys hear me? Can you hear me?
Yes.
Okay. Okay. So who has questions and who doesn't have questions? I'm interested in those who have questions. Your microphones are facing mute. Let me try and ask you again. Just a minute, just a minute. I see raised hands. Let me ask you to unmute. Okay. Okay, you can ask me. Sorry about that. You can ask me. You can ask me. Microphone is now open. You can unmute and ask the question. Your hand is up. Ask.
Madam, I beg your pardon on the second signal of the CD40 self-proliferation.
Of the CD40?
Yes, the second signal.
Okay, others, please mute your microphones as I answer Sewanyana, please, so that we can all learn from this. Others, mute your microphones as I respond to Sewanyana's question. So the second signal of the CD40 cells, after we've had the T cell receptor and interacting with the antigen peptide MHC2 and the CD4 attaching to this for the further group, the second signal comes in from costimulatory molecules or ligands, which are both present on the antigen presenting cell and the CD40 cell. So the antigen presenting cell has two ligands of the B7 family. That is CD80 and CD86. So either of these, not both of them, but one of these interacts with the ligand on the CD40 cell. That is a CD28 ligand. And after these two have interacted together, it's a mere way of binding together. The second is stimulation. Did I answer you? Then yes, after that stimulation, we get to see a pathway of biochemical events happening where the cell starts to secrete cytokines that signal other cells, where it starts to secrete cytokines that affect on the same cell, interleukin 2, which enables the maturation of that cell. Even that second signal stimulates, actually, the first thing it stimulates that second signal, it stimulates the antigen presenting cells to secrete interleukin 12, which affects on a process I call it as paracrine, which affects on the CD40 cell and stimulates it to produce interleukin 2 and also express interleukin 2 receptors. The interleukin 2 this CD40 cell is making comes and acts on that same CD40 cell on the receptor it has done. It has made a process known as autocrine. Now, this interleukin 2 enables the maturation of the T cell. So, Wanyana, is that clear?
Yes, it is very, very clear.
Okay. So, any other question, please?
Yes, doctor.
Yes, please.
Are CD4 and CD8 or part of the T cells, or they are found in blood?
So basically, the T cells are in circulation. They're going to circulation. So you're going to find them. T cells, they are in circulation. They body the different lymph nodes. Are we together? When they encounter the pathogen, that's when they will differentiate to form effector cells specific to that pathogen. Okay, let me try and make something clear. In your body, you have a couple of naive B and naive cells, which are basically died population. But what happens, there are also some effector cells in your body that have been formed in particular to a specific infection you got some days back or some years back. So when you get a new infection, these naive B and the naive T cells, they will differentiate or mature in response to the infection you're having. But not all of them will be activated. Some are activated, and you'll find you have immunity towards that pathogen. Even though you get two different infections, you're going to find different B cells and different cells maturing in different ways, in particular to those different infections you got. I don't know whether I have made you confused or made you even.
No. Okay. No, my question is, when the CD4 and CD8 are coming in as the core signal, my question is, are they part of the T cells that are going to be activated, or they're just free?
So, Jonan, a T cell,
yes,
has receptors on them. A T cell, basically, I'm going to talk about a CD4 T cell in particular, and also talk about a CD8 T cell in particular. So a CD4 T cell basically has both CD4 and CD8 have T cell receptors on them. They are receptors, they're present on the T cell. But what differentiates the two of them, we get to see that CD4 T cell has glycoproteins on its surface, CD8. So all these are on the surface of the cell. While the CD4 has the glycoproteins, CD4. Did I mess you up? The CD8 have the CD8 glycoproteins, while the CD4 have the CD4 glycoproteins. But all these cells have a T cell receptor. Now, these other co-stimulatory molecules, they come when these cells interact with the antigen-presenting cell, which also has its own ligands on its surface. For example, we're going to see that the antigen-presenting cell on its surface, it has ligands or receptors, where we get to see CD80 and CD86, and it also has MHC1 and MHC2. Are we together? So everything you're hearing is on the surface of these cells. Now, when these different cells interact with each other, they interact through these receptors and ligands. Jonan, have you?
All right. Yes, I've gotten it. Thank you.
Yes, the team is raising his hand up or her arm up. Please ask me.
Thank you, Ms. Barbara, for this presentation. Today, I got to realize that I'm having so many figures being thrown at me when you talk about the CD8, CD4, the interleukins 1 to 23, the T helper cells 1, 2, and TH17. What do these numbers signify, and what they based on to give them such numbers? Thank you.
So now, Now, you remember that assignment I gave you in innate immunity when I told you, when I was teaching you about cytokines. I won't lie that I'm going to teach you all the cytokines in one lecture. But like I gave you in innate immunity, and I gave you the different groups of cytokines, I want you to go back to that PowerPoint. Cytokines are signaling molecules, and they belong to different families depending on the signal they make. For example, you're going to find some of them are chemokines because they help in the movement of different immune cells to the site of infection. So you're going to go ahead and see them. Interleukins also differ. Interleukin 1, 2, up, they have different roles they see. Some of them are pro-inflammatory. You're going to appreciate them. So basically the numbers come in depending on the role they do. Are we together? So I want you to go back and do that assignment and get to see the different families of cytokines and appreciate them. When you actually appreciate them, you're going to understand. Are we together?
Yes, we are. Thank you.
Okay, I want another question.
Madam, when are we to hand in the assignment?
No, I don't need your assignment. I want you to read it and understand because I'm going to ask you your exam. And as you know, school opens soon. So you never know, you may come to my exam.
Okay, thank you very much.
Yeah, I need more questions, please. Yes, please. The T cells. So they can actually differentiate into Th1, Th2, Th17, and T regulator cells, depending on the, is that what you want? Some lady speaking Congolese. I love Congolese so much, but please mute your microphone. Okay. Who was speaking? I didn't get your name. Who asked? Okay, Walter. So basically, let me take you through the... So what happens is presenting cell, right? I told you when the antigen presenting cell basically has MHC1 and MHC2. So when this antigen presenting cell is presenting an antigen to CD40 cells, it's going to present this antigen on MHC class 2. But the way it presents it, remember, this antigen presenting cell engulfs the pathogen from wherever it encountered it from, then expresses protein fragments of this antigen on its surface. However, on its surface, there is MHC class 2. I'm talking in particular to CD4. So meaning it's going to have now express an antigen peptide MHC2 complex, which it will present to the CD40 cell. But what interacts with this complex? The receptors that interact with this complex are the T cell receptors, which we find on both CD4 and CD8. But let's stick to CD4. So basically, the T cell receptor on the CD40 cell interacts with this antigen peptide MHC2 complex. After interacting with it, that first signal, we get to see there's also another signal in the first signal, where we get to see the CD4 co-receptors binding to this complex of the T cell receptor and the antigen peptide MHC2 complex. So the CD4 binds to this complex to enable enough binding of the antigen and the T cell. Now that signal is not enough for the T cells, like I said, of the CD4 T cells to get activated. They require co-stimulatory molecules and co-stimulatory signals, which we get to see are provided by the ligands that are present on both the antigen presenting cell and the T cell. So on the antigen presenting cell, we have two ligands, that is CD80 and CD86. Either of these ligands interacts with the CD28 that is on the T cell. Now, when one of these ligands interacts with the CD28 on the T cell, we get to see that actually the T cell gets activated and starts producing a couple of cytokines. But even before it produces cytokines, after the second signal, we get to see that this signal signals the antigen presenting cell to secrete a cytokine known as interleukin-12, which affects on the T cell. I repeat, that is called paracrine, when a cell produces a signal that affects on the neighboring cell, right? Now, when the T cell receives the signal from interleukin-12, it's going to do two things. Number one, secrete interleukin-2 and also express interleukin-2 receptors on its surface. Now, the interleukin-2 it is producing moves out of the cell and acts on the receptors it's expressed on its surface, meaning the interleukin-2 the cell is producing acts on it, a process known as autocrine. And once interleukin-2 has worked on this cell, it actually stimulates the maturation of this effector T cell. Now, this effector T cell can start producing other cytokines like interferon gamma, which is antibacterial and antiviral. We also get to see it stimulating macrophages to come to the site of infection that gets to kill the pathogen. However, for our T cell to become an effector cell, after this first and second signal and the interleukin-2 interruption. So remember, after the first and second signal, we get to see interleukin-2 coming into play and interleukin-12. And the T cell will differentiate to form specific T helper cell. Now, this is why you need to understand. It is at that point when the T cell can differentiate into two different types of T cells, whether Th1, Th17, or T regulator cells. Now, for Th1, like we said, that T cell will differentiate into Th1 when it produces interleukin-2 and interleukin effects on it. We get to see that T cell affecting into Th1. However, in presence of other interleukins, this Th1 this T cell can differentiate to form other T cells. Are we together? You will look through the PowerPoints and get to see in presence of which cytokines does a T cell differentiate to form Th1, Th2 T regulator cells. And all these different cells that T cell forms have different roles they play in the body because they stimulate different signaling molecules that have different effects on the pathogen. Now, for the CD4H T cell, we get to have an antigen presenting cell that is presenting the antigen as an antigen peptide MHC class 1 complex. And this interacts with the T cell receptor of the CD8 cell. After this interaction, we get to see the CD8 glycoproteins attaching to this complex to ensure the stability of this complex. And after that, the CD8 is good to go, unlike the CD4 that requires the costimulatory molecule. The CD8 is good to go. It starts to produce interleukin that affects its own cells, and we get to see it moving into circulation. Whenever it meets a pathogen, it degranulates and releases those granzymes that contain protease, that is bufferin granzymes and granulosin that cause programmed cell death. Did I answer you? How are they formed? I don't know whether I'm understanding it. How they are formed? From the lymphoid progenitor. That is something I took you through at the start. They are formed from the multipotential hematopoietic stem cell. And this stem cell, I told you, divides to form different lineages, where we got to see that it can either be the myeloid lineage or the lymphoid lineage. And in the lymphoid lineage, that is where you get to form the B cells. I mean, you get to form the large lymphocytes and the small lymphocytes. And the large lymphocytes, that's why we have the NAS Are you still hearing me?
No, you had gone off. You had gone off.
Okay, sorry, my internet is really unstable. Let me just try and stay. So I was you, the lymphoid progenitor stem cell divides to form the small lymphocytes. The large lymphocytes are the natural killer cells that play a role
Bob Rayaze.
Yeah, it was.
Yeah, good.
Iwe, Walter, did you hear what she talked about in this MHC1? I think that.
Okay. Yeah. Okay. So I think my internet is really refusing at the end, but I think I've really tried my best with the lecture. I think you can follow after this.
Barbara?
Right? Yes, please.
I want to know about the major histocompatibility complex. You are saying the MHC2 is the one which is on the antigen presenting cells. Then in the activation of the CD8, you talked about MHC1. Can I know where the MHC1 is, or whether it's also on the antigen presenting cells?
I'm going to repeat myself. I'm going to repeat myself. All cells in the body have MHC1. An exception with antigen presenting cells, they have MHC1 and MHC2 on the same cell. So it's basically these antigen presenting cells, we have macrophage, dendritic cells, and B cells. They present both peptides or both receptors on the surface. Are we together?
Oh, I get it.
Me. So you find,
yeah.
Yes. So unlike the normal cells in the body, which use the MHC1 to present whether they're help in differentiation whether they are infected or not, the antigen presenting cells use the MHC1, MHC2 to present the antigen to either CD4 or CD8. So this MHC1 and MHC2 they can actually bind to cells on it. One side MHC2 can bind a CD4, and the other side of the macrophage that has MHC1 can bind a CD8 at the same time. Does that make sense?
Yeah, it does. It does.
Okay. So I really hope we've all registered are clear to you. Is there any other question?
Yes, I have a question, a concern actually.
Yes. Yes, please.
Yeah, it doesn't concern this lecture particularly, but it's about when it comes to our mid-semester exams. We have already covered a lot of work, and I don't know when we are actually having exams or not.
Don't worry, you're going to have exams. Let's first reopen.
No,
of course you will have exams.
Yeah, but then the time when it comes and we have already covered a lot. I just wanted to know if it is going to be mid-semester exams or just like sort of a test.
So remember, you even haven't finished end of semester exams. You are in the middle of them. So number one, when the semester resumes, we shall first finish the practical and the viva. Then we can think of these exams. We need to first finish one semester before we can run to another. The pandemic hit us so bad we couldn't conduct practicals online or viva online. So it's not in the rules of the university to start exams even another semester before you finish exams of the previous semester. So when you guys come back, you will be starting on the exams of last semester. We jumped into another semester so that you guys don't lose time and we don't lose time. Are we together about?
Okay. All right.
So I hope all of us are okay. I'm going to contact the lecturers for next week. Yes, please.
Yeah, I have some questions like what's the difference between positive selection and negative selection? And you talked of thymic hormones. Are there particular hormones that cause the maturation and differentiation of the T cells in the thymus gland.
Now for the thymic hormones, I wanted to go and read about that because it's a couple of hormones involved the same way you see thymic acid selection. I don't know whether you entered later. I feel like I was very clear about that. The cells, that is the CD4 and CD8 that can recognize cells. So basically with primary selection, when a cell identifies the cell peptide, that is the MHC1, and not attack it, that cell is retained by the body, proceeding to maturation. However, when a cell identifies the cell peptide, that's MHC1, that one is killed before it matures. That is negative selection. It's basically a process by which the body eliminates the cells that will help in the immunity, those ones that will actually cause harm in the future. So primary basically selects those that don't identify cells. And negative, we get to delete those that identify the cell peptide, which is the MHC. Did I answer you?
Yes.
Okay. Okay, so I think we can stop there. I already saw that some few slides, 11 slides, and I
Sound is off.
I'm really sorry about my internet, but the good thing the lecture is done. Eh? So I'm going to make available the PowerPoints on the LMS, all of them that you've been missing. You're going to find them on LMS, and make sure all of you have registered. Okay, I wish you a nice weekend and a nice week ahead. I'll contact the lecturers that are supposed to take you through the lectures ahead so that they can prepare. I wish you a nice Sunday. Thank you so much.
Thank you.